Ever wondered how the process of DNA replication comes to an end? Just like any good story, the replication of our DNA has a start, a middle, and an end. Let's delve into the fascinating world of termination in DNA replication. The process of termination is a precise dance choreographed by a host of enzymes. It starts with the removal of RNA primers, the short pieces of RNA that kickstart DNA replication, by the enzyme DNA polymerase. This enzyme, in addition to its role in synthesizing new DNA, also has exonuclease activity. This allows it to snip away the RNA primers, leaving behind gaps or nicks in the DNA. But don't worry, these gaps aren't left open for long. DNA polymerase, in its role as a builder, fills in these gaps with DNA. Once the gaps are filled, another enzyme, DNA ligase, comes into play, sealing the nicks and ensuring the integrity of the newly synthesized DNA strand. DNA polymerase also has another crucial role. It proofreads the new DNA strand, checking for errors, removing them, and replacing them with the correct bases. However, in eukaryotic organisms, those with linear DNA, there's a unique challenge. When the RNA primer at the 5' end of the daughter strand is removed, there isn't a preceding 3' OH end for DNA polymerase to use to replace it with DNA. This leaves a gap at the 5' end of the daughter strands, a missing piece of DNA. This missing DNA could lead to a loss of information, which is a serious issue as we're talking about the blueprint of life here. This is where telomere sequences come into play, filling in the gap and maintaining the DNA's integrity. Telomeres, the linear ends of DNA, contain specific GC-rich repeats, known as telomere sequences. While these sequences don't code for anything, they are essential in solving this end replication problem. So there you have it, the complex yet beautifully orchestrated process of termination in DNA replication. It's a dance of enzymes in DNA, proofreading and gap filling, all leading to the perfect replication of our genetic material. Isn't nature amazing? Now you might be wondering, what is the role of these telomere sequences? To fully grasp this, let's first imagine the DNA replication process. As the RNA primers of both the leading and lagging strands are cleaved out by the exonuclease activity of DNA polymerase, gaps are formed. These gaps are then filled with DNA and sealed by the enzyme DNA ligase. But there's a twist. In eukaryotic organisms with linear DNA, when the RNA primer at the 5' end of the daughter strand is removed, there isn't a preceding 3' OH for DNA polymerase to replace with DNA. This leaves a gap at the 5' end of the daughter strands. If left unfilled, this missing DNA could lead to a loss of crucial genetic information. Enter the telomere sequences. These sequences, found at the linear ends of DNA, contain specific guanine-cytosine-rich repeats. They don't code for anything, but they play an essential role in filling in the gap in the daughter strand, maintaining the integrity of the DNA. As the replication process continues, the replication forks eventually reach the terminating recognizing sequences, also known as TER sequences. These sequences are approximately 23 base pairs long and serve as the binding sites for TUS protein. Once the TUS protein binds to the TER sequences, a complex, the TER-TUS complex, is formed. This complex plays a vital role in the replication process. It's like a traffic cop at a busy intersection, halting the replication forks in their tracks and effectively bringing the process of DNA replication to a halt. So, the telomere sequences and the TER sequences, although seemingly insignificant, play a crucial role in the replication process. They ensure that no genetic information is lost and that the replication process is controlled and orderly. And that, my friends, is how the complex process of DNA replication comes to a complete stop.